Hello and welcome to another installment of very entertaining videos. Today's topic is male reproductive, and there's really two things that I want to focus on as we go throughout this lecture. I want you to think, first and foremost, about this being a continuation of our discussion on endocrinology. We'll build on some of the ideas that we presented in endocrinology, including the hypothalamic pituitary axis and its role in regulating male fertility. The other thing that I'd like you to think about before we get started is that much of what we talk about today is also applicable for female reproduction. As we detail out female reproduction, specifically folliculogenesis in another video, I want you to look for parallels between the two systems. So let's build on where we kind of left off with endocrinology, going back to the hypothalamus. There's our hypothalamus. We've got our infundibular stalk leading out to the pituitary, the anterior pituitary, the posterior pituitary. And just to reiterate this idea of the hypophyseal portal system, the hypothalamus is releasing gonadotropin-releasing hormone, GnRH, and this is traveling through the portal system. Remember, portal system, two capillary beds in series. A capillary bed here to pick up the GnRH, and then another capillary bed here to drop the GnRH off, and that GnRH will target cells on the anterior pituitary, causing the release of two hormones. And these hormones, LH, luteinizing hormone, and FSH, follicle stimulating hormone, will enter the capillary bed and then travel throughout the blood to their target organs. This is endocrine signaling, right? This is signaling through the blood. Now, you might be surprised as we look at LH and FSH. Obviously, these were named for their role in female reproduction. Luteinizing hormone actually refers to the color yellow. FSH, follicle stimulating hormone, folliculogenesis, this is all female reproductive. But we see an essential role for these hormones in male reproductive as well. And again, building on this theme that there are a lot of similarities. All right, so from the anterior pituitary, we have LH, we have FSH. And while the two work together, they have different targets and they're actually regulated independent of each other. The target for LH is a cell in the testes called the Leydig cell. And we'll detail some of the anatomy here in just a second, but for just the signaling, let's think about it in terms of arrows and functionality. The Leydig cell is the cell in the testes that produces testosterone. And not surprisingly, testosterone is the key negative regulator. So testosterone will feed back and inhibit specifically LH from the anterior pituitary, and to some extent it will also inhibit GnRH release from the hypothalamus. All right, switching over to FSH. The target of FSH is a cell, again in the testes, called the Sertoli cell. And very similar to follicular cells in female reproduction, the Sertoli cell has the role of regulating sperm maturation. You can think of Sertoli cell as the guardians of the sperm. Now, it's true that testosterone is a key player in this process. And these Sertoli cells have a lot of androgen-binding proteins that help sequester the testosterone that's produced right around them, and they use this testosterone to drive sperm maturation. However, testosterone is doing a lot of other things in the body, and we want a way to uncouple testosterone from sperm maturation. And this is shown in feedback on FSH through a protein called inhibin. So Sertoli cells will make and release inhibin into the blood, and inhibin specifically comes back and targets FSH and blocks FSH from being released. But it doesn't deal too much with LH or GnRH. This is how we can isolate and separate the regulation of these two hormones, LH and FSH, even though the expression of both of them is dictated by GnRH from the hypothalamus. All right, now that we have the basics of the endocrine signaling and the hormones involved in spermatogenesis, let's first take a look at the anatomy. By understanding the anatomy, we can better understand how to get from point A to point B and really the whole story of what's going on. Of course, our end game is the production of sperm through a process called meiosis, and then the storage of that sperm in anticipation of an ejaculation into the female vagina, where the journey begins for the sperm in the hopes of achieving fertilization. We can describe male reproductive anatomy with a fairly simplistic picture. Here, from a macro view, we can let's orient ourselves. We get the testes. Here we have a structure called the vas deferens, also known as the ductus deferens. This proceeds on the anterior or ventral surface of the bladder. You can show the bladder right here. The vas deferens swings up and over the bladder and then around on the the posterior side where it encounters the first major contributions to ejaculate in the form of seminal vesicles. These are paired vesicles back here. They're going to pass through the prostate. This is where the ejaculatory duct merges with the urethra. Then there's one more set of glands, the bulbourethral glands, that come into play here more distally. So let's zoom in and let's kind of take a look at this. The first structure we want to look at in detail is the testis, highlighted in the boxed region here. Let's zoom in on this and label some of the key anatomical structures. So we've got, down here shown in blue, the seminiferous tubules. And this is actually the site of spermatogenesis, which we'll highlight in more detail in just a minute. Following spermatogenesis, sperm are collected in this structure here called the ready testis, where they're then passed on to this structure, the epididymis. The ready testis is all about collection, and the epididymis is all about maturation. The epididymis is actually quite long, anywhere from 6 to 7 meters. That's pushing about 20 feet in length. And the sperm could hang out here anywhere from 3 to 5 months as they finalize the maturation process. And then upon ejaculation, they travel through this structure here called the vas deferens. Now, as the key topic of this video is about spermatogenesis, let's jump back down here and let's take a look at what's going on in the seminiferous tubules. What we want to do is kind of take a cross section through this tube and see what it looks like. All right, here's a cross section of the seminiferous tubule. And as you look at this, there's a couple of things that I want you to notice. Number one, it's a hollow tube. The lumen, this is where we're going to collect sperm as they're created. And then we've got 3 cell types that we want to talk about. First, these green cells here surrounding the tube, these are the Leydig cells. And remember, their job is to produce testosterone. They're under the control of luteinizing hormone from the anterior pituitary. And the second cell type, shown here in purple, this is the Sertoli cell. And we call these cells the guardians of spermatogenesis. They're going to facilitate and regulate sperm production. Now, how I've got this drawn out, and it's not like this anatomically, but it helps us understand the process of spermatogenesis. I want you to think about this as a clock, where if this is the clock face, we're going to follow the process of spermatogenesis in a clockwise fashion, and then we're going to see that from a spermatogonium to the creation of 4 released spermatozoa into the lumen, we're looking at about 60 days. And I also want you to appreciate that the journey that these spermatozoa will take during their creation is a journey from the outside toward the lumen on the inside. So let's detail this journey. We start with this cell right here called a spermatogonium. This is a derivative of an embryonic cell called a primordial germ cell, or PGC, and this cell is diploid, 2N. Now, what do those terms mean? Diploid, di for 2, tells me that I have my full cohort of chromosomes. Because humans are sexually reproducing, this means that I have a set from mom and a set from dad, hence di, diploid. And for humans, this is 23 chromosomes from mom, 23 chromosomes from dad. Diploid for a human is 46. The 2N, this tells me my total DNA content. In other words, I haven't started replicating. I haven't copied any DNA. This is our beginning point, and our next step, as we proceed clockwise, again, set up like a clock. Not that we're moving clockwise, but just to keep in mind the concept of time. The cells, you can see, are kind of squeezing in between these Sertoli cells, the spermatogonium, as they differentiate. And this next cell down here, still just one cell, this is a primary spermatocyte. Now the difference is that the spermatocyte is diploid, but now it's 4N. In other words, we've replicated the DNA in preparation to begin meiosis. In other words, this is moving essentially from G0 through G1 and S phases of the cell cycle, and this takes about 16 days. All right, as we continue the process, in this next area, you're going to recognize we have 2 cells. In other words, we've undergone our first cell division. These cells are called secondary spermatocytes. To get from primary spermatocyte to secondary spermatocyte, we undergo meiosis 1. This takes about 8 days, and it's in this process where we see homologous recombination. This helps build diversity into the genetics of these newly created cells because this is a physical mixing of mom's and dad's chromosomes. So as chromosomes come out the other end, they're part mom and part dad. And because we've undergone a division, these cells are now considered haploid in the sense that now each cell has 23 chromosomes instead of 46, but also recognize that that chromosome is still really two sister chromatids together, and so this is considered 2N, where our final destination will be 1N, a haploid 1N cell. All right, we undergo then another cell division. We now have 4 cells. These are called late spermatids. These are haploid 1N, and we get there by the completion of meiosis II, which takes about another 16 days. And then our final product, released into the lumen, the spermatozoa. This maturation stage is called spermiogenesis, and it's roughly 20 days, completing the roughly 60-day cycle from beginning to end. Now, as we kind of zoom back out, coming back and looking over at the seminiferous tubules, you recognize that this is just one place along multiple seminiferous tubules, all asynchronously producing sperm. And following the onset of puberty, this will continue throughout the life of the adult male. Of course, maximal production is probably in the early 20s, and then it wanes off during the lifespan, but production is always happening. And on average, we see production at a rate of about 5 million per gram of testes, with each testis weighing about 20 grams on average. This gives us about 100 million sperm per testes per day. All right, so now that we've got sperm creation, let's take a step back and finish the journey. If we come back to our image of male anatomy, let's take this structure and let's just straighten it out, starting with the vas deferens. So we're starting on this end with the vas deferens, and we first encounter the paired glands called the seminal vesicles. We stated earlier that this provides the bulk of the ejaculate. This is about 60% of the total ejaculate by volume. And things that we're going to find in here: fructose, citric acid, things like fibrinogen, prostaglandins. Of course, you would recognize fructose and citric acid as energy-related molecules. Fructose can run glycolysis. Citric acid goes right into citric acid cycle. These are driving mitochondrial production of ATP in significant ways, and ATP is required for the swimming motion, the flagella of the sperm to swim. Fibrinogen, we saw this in clotting, and this is important for the ability of sperm to adhere to the vaginal and uterine walls as they swim, so they're not washed away by vaginal secretions. Then prostaglandins, general signaling molecules. From this point down to the prostate, we call this the ejaculatory duct. And then, as we mentioned, the prostate, it's a fairly large gland. This is at the junction of the ejaculatory duct and the urethra coming from the bladder. And the prostate contributes significantly to the ejaculate. This is another 30%. And this is all about buffering. This secretion contains an alkaline pH that is necessary to combat the very acidic pH of the vagina. Shortly after the prostate, we encounter the bulbourethral gland. This contributes another 5% to the semen, and this is all about mucus for lubrication. It's fairly easy to identify in that it's just downstream of the external urethral sphincter shown here. This portion of the tract is called the membranous urethra, and the last portion of the tract, the spongy urethra. And a little bit of math tells you that only about 5% of the total ejaculate actually came from the testes through the vas deferens containing the sperm. Most of the ejaculate from the seminal vesicles, and then the prostate, and then a little bit from the bulbourethral gland. All right, the last thing that I'd like to do is just provide a quick anatomical description of a sperm cell. We begin with the head, then we have what's called the midpiece, and we finish with the tail. The head, in addition to containing the genetic material, which remember, would be at haploid 1N, also contains a structure called the acrosome. The acrosome contains hydrolytic enzymes that will be necessary to get through the zona pellucida that surrounds the oocyte in the process of fertilization. The midpiece is chock full of mitochondria. And of course, this is all about ATP production, necessary to drive the microtubules that make up the tail that cause the flagellum to move. And with any cell, fitness is important, and selection is happening even through the process of intercourse. Some of the faster swimming sperm can make the journey from ejaculation to the ampulla of the fallopian tube, where fertilization is ideally to take place, in 3 to 4 hours. Other more endurance-based sperm could make the journey up to 24 hours. And we'll talk about that process of fertilization in another video.